Okay, hello, 12s. So we're on to Unit B, enzymes and metabolism, and some of this we've already covered in Unit A. So this is gonna be awesome. So let's start first. Unit B starts with us talking about enzyme structure. So remember, we talked about protein folding, how proteins fold, and they have those different levels of structure. Primary, which is the order of amino acids. Secondary, which is the beta pleated sheets, and the alpha helix, which is between the amine group and the carboxyl group, those hydrogen bonds. And then there's the tertiary structure, which is all those R groups, right? All those, the things sticking off. They have like sulfide bonds and hydrogen bonds and ionic bonds and covalent bonds with each other, and that forms and shapes the protein. Well, enzymes are proteins, so that tertiary structure is super important to the function of an enzyme. It makes the shape of the enzyme, and also quaternary structure, because two peptide chains can come together and make a shape. So enzyme structure, super important. The 3D shape is super important. Sometimes this thing spins around and makes like a cool thing. I don't know what it is, but it's an enzyme in 3D shape. So what enzymes do is they lower, this is very important, lower the activation energy of chemical reactions. Remember, we talked about chemical reactions as those electrons interacting with each other. Chemicals, atoms sharing electrons or giving up electrons and taking them away. That's a chemical reaction. So in order to get those chemical reactions, though, those atoms have to bang into each other. They have to have enough energy to get close enough together so that they can rip off those electrons and share them, or the electrons can come flying off, they become ions, and you make ionic bonds. So there needs to be enough energy in the system to do that. So a lot of chemical reactions, you need to heat them up in order to have the chemical reaction happen, because you need to get those atoms moving fast enough to smash into each other. But that poses a problem for biological organisms, because if you heat them up, if you catch them on fire, they don't work very well. That's not good. We don't want to be on fire. So we have enzymes, and what enzymes do is they grab the atoms, or they grab the molecules that you want to join together, and they just physically smash them together. So the activation energy is lower. You don't need all that heat or that energy or whatever, electricity or whatever you need. It'll be heat to get it going. So the enzymes lower it. And so you can see that the structure, the 3D structure, is really important, because this 3D structure of the enzyme and those properties of that enzyme, of those R groups, whether they're charged, they're not charged, they're polar, non-polar, will determine how they grab and what they grab. Okay? So that's why the 3D shape is super important. So an enzyme will have what's called an active site. And that is, like if you imagine my hand is an enzyme, and the active site is going to be like this part, the part that grabs stuff. Okay, this enzyme grabs this. It doesn't do anything with it, though. It's not a very good enzyme. But if I had a little phone stand here, okay, watch this. We're getting really terrible here. Okay, imagine this is an enzyme. Okay, this is the active site, and it grabs two things and puts them together, and then lets them go. That's basically what enzymes do. This one doesn't work very well. It's got a mutation, so it doesn't work very well. But it puts things together. Or it can grab things and take them apart. Okay, so the 3D shape is really important. So this part is specific for this. It's specific for this Contigo lid. And this part of the enzyme is specific for the Contigo body. They can't do anything else. So an enzyme will be specific, the shape of it will be specific for the substrate. So what an enzyme does is it takes a substrate and then produces a product. And that product might be, it breaks things apart, or that product might be that it puts things together. Okay, enzyme. Okay, so now enzymes have we're just going to talk about the name here. So the name of an enzyme is usually the substrate followed by ACE. So it's usually the thing that it's specific for, followed by the word ACE. So our enzyme that put that thing together would be contigo-ACE, right? This is its substrate is the this contigo container, so it will be just called contigo-ACE. If we had an enzyme that ripped paper, it would be paper-ACE. The name is not the product. And this is, sometimes this doesn't, sometimes, like what do they get? Maltase breaks up maltose into 2 glucose molecules. There's a total awesome enzyme product thing. So because the name is not gluco-ACE, because that's the product, it's maltase because that's the substrate. Keep that in mind, okay? Enzyme-substrate complex. What does that mean? An enzyme-substrate complex is when the active site joins with the substrate. So when it binds to a substrate, they become an enzyme-substrate complex. It's just a temporary arrangement. This just happens for a moment while, the product is being made. And so the chemical nature of those R groups, whether they're polar or non-polar, is what will determine the substrate will be attracted to the active site and form that enzyme-substrate complex. But once it forms the enzyme-substrate complex, the reaction happens, and then it's no longer attracted to the active site because it's no longer the substrate, it's the product now. It's been changed. So maltase is attracted to maltose, the sugar, and then the sugar jumps into the active site, and the shape of the enzyme and the chemical nature of the active site breaks maltose into 2 glucose, and it's no longer attracted to it because now it's not maltose anymore. It's glucose. It's been changed. It's sad. And then the enzyme returns back to its original state, and it can grab another maltose. So the enzymes are not used up in the chemical reaction. So that is important. They're not used up. They can do it again and do it again and do it again and do it again until they break. They do wear out. Okay, so this video might be a little bit long. Anabolism versus catabolism. Well, we learned about this. So dehydration synthesis is when smaller molecules are merged to form larger molecules, right? So that's anabolism. It's endothermic. It requires energy. We talked about that. And catabolism is the reverse. So it's when you break things apart, and it's exothermic. It releases energy. A great example is when you break down ATP to ADP. You break that bond, those phosphate bonds, that high energy bond. So an enzyme does that, and it releases heat. It releases energy. There's a thing in the book where it says they can go back, but one enzyme, I looked it up, the reverse catabolism, but it is reversible, but it's not going to be the same enzyme that does it. Let's just stick to that, because then the name of the enzyme would change. Let's just leave it at that. But the reaction is reversible, so you can break that phosphate bond, and then you can put that bond back. You can break sugar apart, and then you can put it back. You can take glucose molecules, take them apart. You can take maltose apart into 2 glucose, and then put them back together. There's a video in the notes. It's me talking. It's probably me saying the same stuff that I'm talking about here. I have no idea what it is. I have not watched it. I don't know. I doubt there's anything good in it. Okay, so we've covered B1. I think by the end of today, according to my schedule, we're supposed to cover B2. Sorry, gang. So this video is longer. All right, for some reason, I didn't put them all together. All right, let's look at B2. B2, give me some B2 action. Okay, B2, models of enzyme activity. So the important one here is, lock and key and induced fit hypothesis. So lock and key states that the enzyme's active site is like a lock, and the substrate is the key. And the substrate only fits in one specific way, and then it unlocks the reaction. So everything is static in this model. Nothing really changes. So there are these rigid things that fit into each other. The induced fit hypothesis is another hypothesis, and this is, now we know that this is most likely the right one, is that the things biology is never rigid. Things aren't rigid. And so things are a little bit more flexible. So as the substrate comes into the active site, those chemical properties, the electrochemical properties, so, like the charges and stuff, will change the shape of the enzyme. It might squish the substrate and change the shape of the enzyme. So it's kind of a more gradual, and that leads to the reaction, because all of a sudden now, that enzyme, maybe hydrogen bonds are brought closer together, or covalent bonds are stressed, and that as the substrate comes in, and it wants to break open again and smash the substrate, something like that. So this model is more true to life. The other thing we need to talk about is cofactors. And so Apoenzyme, that word, is just the peptide, it's just the protein of the enzyme. Holoenzyme means everything. And so what happens is there's these things called cofactors. So enzymes often don't just work on their own. They need to be activated. You need to eat vitamins like vitamin B and C and that kind of thing, because they're minerals that act as cofactors for important enzymes in your body, which means that, so you've got the peptide chain, you've got the active site, but it's incomplete. And so something has to come and attach to it to make it complete, and that's these cofactors. So that it becomes a holoenzyme, it becomes holistic, right? Holistic means the whole. We're doing everything. We're treating the spirit, the mind, and the body. An apoenzyme is we're just going to cut out the thing that's broken and put it back in. It's just the protein part. So there's a nice picture here. This is the apoenzyme. That's the protein part. There's the substrate. There's the cofactor. The substrate can't bond with the apoenzyme until the cofactor comes in. So the cofactor comes in. Now the substrate can bond, and the reaction can happen. There's two kinds of cofactors. So there's metal ions like potassium and zinc, and they've got like a lot of charge. So again, they affect everything. Here is these chemical properties. They got a charge. They affect the shape of the enzyme. They react with those R groups, right? And then another one is it can be actually just another. It can be a protein or vitamin. So NAD takes up excess hydrogen, some electrons during cellular respiration, so it helps balance things out and produce ATP. So that's why vitamin B is really important, because cellular respiration, as you know, is super important. Okay, so we'll end it there. Go over those notes. There's another video here. I have no idea what I say in it. Maybe I say some totally different things than I just said now. Who knows? I might contradict myself. I hope not. I don't know when that video was made. I think it was made earlier this year or last year. I don't know. Or I stole it from when I taught this in summer school. So let's just leave it at that. That is we're starting unit B. That was B1 B2, models of enzyme activity and the shape of enzymes. We're going to fly through this stuff because we have a short week, and this stuff is sort of a repeat of what we just did. Okay, cheers.